Hello everyone, welcome to the General Microbiology Lab Session 7. In this video, we will be discussing about microbial characterization techniques 1. And one of the most popular techniques that has been widely used in the worldwide is microbial staining. In this video, you will also learn about the procedure, how to identify the properties and characteristics of bacteria using gram staining. So the learning objectives of this presentation is to obtain the properties and characteristics of a prokaryote culture and also differentiate between gram-negative microorganisms and gram-positive microorganisms. Just to refresh from what we have learned, that the major shapes of bacterial cells are involving rod-shaped or bacilli, circular or spherical that we call it fasciae, and spirals. They can be found also as single cells like Monococcus or Monobacillus. They can also be found in pairs, for instance Diplococcus or Diplobacillus, clusters of 4 cells which we call it tetrad, 8 cells we call it Hartina, and irregular clusters that we call it Staphylococcus. That will be explained in this slide. There are actually various methods for classifying microbes, such as morphological characteristics, differential staining that we will conduct in this presentation, biochemical staining, polymerase chain reaction, and DNA chips. One of the most common staining procedures is the gram staining. As we can see from the picture, that Cossi has round or circular shape, and bacilli has actually rod shaped, and others like spiral shaped or coma shaped for Vibrio cholera. The Cossi also it will be divided into several types like Coccus, Diplococci, Streptococci, and Staphylococci. And for the bacilli, it is also divided into several types that we can call it Streptobacilli, which has chain shape, and then Diplobacilli. Each of these cells have organelles within the cells, and the staining method itself can affect different parts of the bacterial cells, including the cell walls, capsule, flagella, and spores. Here are the structures of gram-positive cell wall and gram-negative cell wall. As you can see it clearly that the gram-positive bacteria have thick peptide glycan, and the gram-positive cell wall consists of membrane proteins, cytoplasmic membrane, and thick peptide glycan. Meanwhile, gram-negative cell wall consists of outer membrane, thin peptide glycan, and cytoplasmic membrane. So, the peptide glycan is actually the key as we will stain or color this part during the experiment. Before we jump to the staining method, I would like you to understand the parts of the cell once again. So here is the link to understand the characteristics of bacteria. Finally, we are in the main core of the presentation. So the gram stain is a differential staining technique that differentiates bacteria based on structural differences in the cell wall. The bacteria that have a thick peptide glycan layer, we call it gram-positive, and by the end of the experiment, it will have purple color. Meanwhile, the bacteria that have a thin peptide glycan layer, we call it gram-negative, and by the end of the experiment, it will show us pink color. For better understanding the gram stain technique, I have also prepared to you the link that you can access from your own PC. After watching the video, you will find out some reagents were used in the gram stain technique. And these are the reagents used in the gram stain technique. The first one is the crystal fiolate, which is used to color both gram-positive and gram-negative bacteria, which is resulting in the purple color for both cells. The second one is iodine. The iodine acts as a dye fixator that fixes the purple stain in both gram-positive and gram-negative bacteria. And the third one is 95% ethyl alcohol, or even ethanol. It is actually used to decolorize the gram-negative bacteria as it dissolves the outer membrane and the thin peptidoglycan in the gram-negative bacteria. And the last one is safranin. Safranin is used as a counterstain. It acts actually like a crystal violet, but it is specifically staining the gram-negative bacteria, which resulting in the pink color. So students, in this slide I would like also to explain to you the procedure how we start with the gram stain technique. There are actually 3 steps to start with the gram stain method, and the first one is making smear. So firstly, we have to ensure the slides are clean. Make sure the slide that you have is clean. If the slides are dirty, we have to clean the slide with clean water or ethanol and dry them with the tissue paper. I hope this is quite clear. And the second one, we have to place a loop of water on the middle of the slide, as you can see from the picture. And then we can aseptically add bacteria to the water using tip of the needle. And make a smear by mixing the bacteria with water in a circular motion. The smear should be slightly cloudy, as it means that the bacteria has mixed with water. And then make a smear by mixing the bacteria with water. After mixing the bacteria with water, we can let it dry. Also, to hasten the process, the slide can be held high above a flame, not touching the flame, just high above a flame. However, there is also a risk that this process might disrupt the staining process of some gram-positive samples. And then we can heat fix the smear by moving the slide several times, 2 to 3 times through an open flame. Please avoid overheating. Use a pair of tweezers if you can't stand the heat. Okay, and then now the sample is ready for gram staining. I hope this procedure is quite clear to you for the first step. After making smear, now the samples are ready for gram staining. In the gram staining method, firstly we have to cover the smear with 2 to 3 drops of crystal violet and leave it for 30 to 60 seconds. Tilt the slide downwards into a waste beaker and rinse it with water and gently tap the remaining water with a tissue paper, as you can see from the first picture. And then the second picture showed us that we have to add 2 to 3 drops of Lugol solutions or iodine solution on the smear for at least 60 seconds, as we can see from the second picture in this presentation. And again, we have to tilt and rinse it with water and gently tap the remaining water with a tissue paper. The next step, we have to decolorize the smear with acetate alcohol solution. After the runoff is clear, quickly make a thorough rinse with water. Do not let the smear stay too long under the acetate alcohol solution, or a gram-positive sample will appear as a gram-negative. And finally, we can add 2 to 3 drops of a counterstain, which in here we use safranin as a counterstain, and wait for 30 seconds. Tilt and rinse with water, and gently tap the remaining water with a tissue paper. And after that, the samples are ready for microscopic observation. And finally, we are in the last step of the gram staining method, which is called microscopic observation. After making smear process and gram staining process, the last one is microscopic observation. Firstly, we have to put the slide on the microscope stage, and we have to start from the lower magnification lens, adjust the focus, and find the desired area. The bacterial cells should not be too concentrated. Usually, cells at the margin of smear will be less clumped together. And then add a drop of immersion oil before using the 100 magnification lens. Do not let the 40 magnification lens touch the oil. This can be achieved by two ways. First, from 40 magnifications, tilt the revolving nose piece and add the oil when the sample is located in between the 40 and 100 magnification. Or rotate the nose piece to the 4 magnification and add the oil to the sample. Move directly to the 100 magnification from 4 lens. Once the oil is added, shouldn't return to the 40 magnification. Furthermore, you don't need to lower down the stage when adding the oil as it will change the focus. And finally, we can observe the bacterial shape and color as you can see from the picture. We can see that there will be a gram positive and a gram negative. For the gram positive, the color will be purple, and for the gram negative one, the color will be pink. And the example for the gram positive is Kasi, which has round shape or spherical. Meanwhile, for the gram negative one is Basili, which has rod shape. And after that, you have to record your result of observation. These are the references that I used during the experimental study. If you have any questions or suggestions, feel free to contact me. And thank you very much for your close attention. See you in the next lab. Bye bye.